Another type of movement is called transcytosis. This is also bulk movement, and this is used by some cells in the apical basolateral sorting chain or membrane proteins. If you remember, this is the apical part of the cell, and this is the basolateral. Basically, the apical is where materials come in, and the basolateral is which is attached to the extracellular matrix. So this process of transcellular transport, which combines endocytosis and exocytosis, can be employed to import extracellular ligand from one side of the cell and transport it across the cytoplasm and secreted from the plasma membrane at the opposite side. So in transcytosis, you have bidirectional movement. Materials can come in from the basolateral side and be moved all the way out, or materials can come in from the apical side and through the vesicle formation be moved to the basolateral side of the protein of the cell. And that is true especially when you eat food. Remember, your gut is lined with these epithelial cells. So when you eat food, it is absorbed through the apical membrane. It goes through, and down here you have the blood vessel which takes up material. So there are three common pathways of exocytosis. Exocytosis in cells involves the fusion with lysosomes, and it is called this exocytosis which happens in cells, and this involves fusion with lysosomes, which is what we've just gone through. And then there's constitutive exocytosis. This is the regular secretion of molecules. And then there's regulated exocytosis. So the three types, the one which involves fusion with lysosomes, constitutive exocytosis, this is for secretion of molecules which have been made inside the cell. And also there's a third type called regulated exocytosis, and we'll talk about all three in individual. So pathway number one is exocytosis in cells, which involves the fusion with lysosomes. These are the organelles which contain acid hydrolase enzymes, and these enzymes are known to break down waste materials, microbes, and also cellular debris, used material from the cell. So lysosomes carry their digested material to the cell membrane, where they fuse with the membrane, and then they release their content into the extracellular matrix or the extracellular fluid. The matrix is found on the basal side of the cell, and the extracellular fluid is outside the cell on the apical membrane. Pathway number two is constitutive exocytosis, and in constitutive exocytosis, it involves regular secretion of molecules, and this is an action which is performed by all cells, irrespective of where they are. Constitutive exocytosis delivers membrane proteins and lipids into the cell's surface, and once there, it may expel some substances into the cell's exterior. So it depends on where that molecule is going. Remember, we talked about materials being made in the ribosomes going through the Golgi, being encapsulated in vesicles and going to the plasma membrane. So they go to the exterior that way. Then pathway number three is regulated exocytosis. This is special because it relies on the presence of extracellular signals. This is signals that are outside the cell. And when these signals bind the cell, you get expulsion of materials within the vesicles. This is really special because things like insulin are made and stored inside the cell. And once the pancreatic beta Langerhans cells, for example, So there's a signal which comes and binds the surface receptor of that cell. And when it binds that surface receptor, some signals are released into the cell, which say, can you release some insulin? And it is only that time, it is only then that you get expulsion, the vesicles binding and releasing the insulin into the ducts and out into circulation. So regulated exocytosis will occur commonly in secretory cells. I've given you the pancreatic beta Langerhans cells. You can also have the thyroid cells. You can also have the pituitary cells, which release protein hormones. So there are a whole variety of cells in the human body which release proteins into circulation. And I believe you did many of these during physiology. So it happens in secretory cells, but not all cell types. So cells which are not, which do not secrete materials, do not have regulated exocytosis. Secretory cells also store products such as hormones, neurotransmitters. These are the chemicals that transmit nerve impulses along the axons, digestive enzymes, and are released only when they are triggered by these extracellular signals. These signals come from outside the cell. Secretory vesicles are not incorporated into the cell membrane, but they only fuse long enough to release their content. So they come, bind the plasma membrane, open into the exterior, release what they have to release, and go back. Once the delivery has been made, the vesicles reform and return to the cytoplasm. So the vesicles are not part and parcel of the plasma membrane. They have a job to do to deliver cargo. It's like a plastic bag. When you bring it into your house and you unload your groceries, you don't keep it in your house forever. It becomes trash. So in certain cells, the secretion of specific proteins is also not continuous. These proteins are sorted in the trans-Golgi network into the secretory vesicles that are stored inside the cell, waiting for the stimulus or the signal for exocytosis. Such regulated secretion occurs in the pancreatic acinar cells, which secrete precursors of digestive enzymes, and also in hormone-secreting endocrine cells. The release of each of these stored proteins, as I've said before, is initiated by neural and hormonal stimuli. In most cases of regulated secretion which have been studied so far, up to 2021, there's always a rise in cytosolic calcium concentration, and this increase in calcium concentration is induced by binding of the hormone to its receptor. Once it binds the receptor, you have a triggering of fusion of the secretory vesicle membrane with the plasma membrane. So first you have the signal or the stimulus. Once you get the stimulus, you get increased calcium. Once you get the calcium increase, you have vesicle moving to the membrane, to the plasma membrane, where it fuses, and then it ejects and releases the contents. And this is by exocytosis. Are we together? Okay so far? You need to tell me if you got lost. Okay, thank you, Daniel. So, what is this special thing about cytosolic calcium? It is essential in both endocytosis and also exocytosis. So let us look at this. Here you have the early endosome, In this early endosome, you have a budding of a portion which contains certain molecules. So when it buds, you get, say, for example, neurotransmitters getting inside and it is refilled. Okay? This, once is refilled in the synaptic vesicle, it is translocated to the membrane. Do you know what a synaptic vesicle is? Synaptic vesicles occur in neural cells. So you can have a synapse between a nerve cell and a muscle cell, and you can also have a synapse between a nerve and a nerve cell. So once it is filled with whatever needs to be removed, you get it docking on the membrane. At the place where it docks on the membrane, in the example of the synaptic membrane, it's called the active zone. This, of course, is the plasma membrane, and once it has docked, ATP comes in, and once ATP comes in, there is priming which happens, and when priming occurs, calcium ions are activated. How are calcium ions activated? Remember the channels we talked about earlier? They open up and release calcium into the cell. You see this calcium? And once this calcium is released, then the material or the neurotransmitter is released into the synaptic cleft. In the case of a synaptic membrane, you have the presynaptic part here, and this is the postsynaptic here. So basically what this neurotransmitter is doing is moving and binding neurotransmitter receptors on the other side through the cleft. This is the space, activates these receptors, and the nerve signal is propagated to the subsequent cells. The same way, calcium, which comes in through this calcium channel here, opens up, causes endocytosis, materials come in, you have translocation, and then you have protons entering the vesicles, and then the protons cause either refilling of vesicles or they move to the early endosome. So calcium is critical in both release of materials from the vesicles and also in endocytosis, in both of them. So if I were to ask you to explain the role of calcium in endocytosis and exocytosis, this is the story that you would probably be giving to me. I would probably ask you, not necessarily in a nerve cell, I would probably ask you cellular cytosolic, or the role of cellular cytosolic calcium concentration in endocytosis and exocytosis. It's up to you if you chose to give this same example, but I'm expecting that you go and read what happens in other cells. So a newly formed cis-Golgi vesicle with its luminoprotein cargo, I'm hoping that you know this cis here is the one which is facing the rough endoplasm, the end of the Golgi apparatus. With its luminoprotein cargo will progress from the cis phase to the trans phase of the Golgi complex, and then into the trans-Golgi reticulum. The reticulum is that part where you have very many vesicles containing the protein cargo. There are very many small vesicles that move proteins from one Golgi compartment to the next, but they do so in the reverse, or what is referred to as the retrograde direction. These vesicles will retrieve endoplasmic reticulum or Golgi enzymes, the ones that don't need to go anywhere else in the cell, but need to be localized either in the endoplasmic reticulum or Golgi to an earlier compartment in the secretory pathway. This is the way that enzymes that modify secretory proteins come to be localized in the correct organelle. So rather than going from cis to medial to trans, they are immediately put in vesicles and they're taken back where they work, either by reverse or retrograde movement of the vesicle. So that is another example of how bulk movement occurs. So how is this? This is the intracellular part of the cell. This is the extracellular part of the cell. This is the outside. This is the inside. Here you have the Golgi apparatus, the trans-Golgi network as it is known. Here you have the vesicles pinching off. Maybe they contain either hormones or neuropeptides or many other things. And this is kept in reserve inside the cell. It doesn't go anywhere until a stimulus comes in, like I showed you before. Then you get ATP and calcium coming in through the channels and then causing secretion. So when these vesicles are ready, they sit inside the cell, and the reason they sit inside the cell is because you cannot afford the delays in transmitting signals, either through hormones or neuropeptides, especially in neuropeptides, because remember the nerve signals need to be moved within milliseconds. If the nerve signals are not moved within milliseconds, if you step on a hot charcoal, it will take forever for these proteins to be made and then to be put in vesicles, and then after being put in vesicles to be released. By the time all that happens, your foot will be burnt, but they are always sitting there ready. And that's why when you step on hot charcoal, you remove your foot immediately, instantaneously, because these things are ready. They come out, and quickly the signal reaches the brain and tells you, can you get that foot off the charcoal? So they come and they sit on the membrane. This is what biologists call kiss and run. Don't ever say biologists don't have a sense of humor. So they release it. Once they release it, it closes and goes back and sits there. So it releases what was asked for. Cavity capture is it takes back what is not required, because remember these vesicles contain a whole bunch of proteins. So cavity capture is just releasing what was asked for by the stimulus, closes off and goes back. Hence the name kiss and run, followed by cavity capture. Full fusion is if everything in that particular vesicle is being released. So it comes, binds, and releases everything. Now, in clathrin-mediated endocytosis, we've gone through this again, the coated pits. And they take in material, pitches off, comes in, links with the endosome, and goes in. Either goes back, retrograde, for those materials that are needed here, or if it is material that need to be degraded, they are shuttled or shunted through vesicles to the lysosomes, where the hydrolytic enzymes do their own work. And that is when bulk endocytosis happens. Okay. So the plasma membrane glycoprotein mature via the same pathway as continuously secreted proteins. I think we did this slide before. I just put it in just to remind you. The maturation pathway taken by continuously secreted proteins is also followed by membrane glycoproteins. Remember the integral membrane proteins that are stuck into the plasma membrane. So newly made glycoproteins are inserted into the rough endoplasmic reticulum membrane, and then subsequently they move through the Golgi cisternae en route to the plasma membrane. And these glycoproteins also have been shown to undergo the same types of modifications in the same endoplasmic reticulum and Golgi compartments that we find secretory proteins. Yes, Leah. Leah, you've raised your hand. Can I help you? Leah? Okay. And of course, we went through transcytosis. This is the apical part. Here you have a protein. Follow the protein in green. Yeah, the basolateral membrane. goes in and it is sorted, and once it is sorted, the receptor goes back and the rest of it goes through the other side, and it can be released in the apical plasma membrane of the cell. On the other hand, apical proteins, which are here, can be internalized by endocytosis. They come in and they can be recycled, or they can actually go out through the basolateral side of the cell. So that concludes our bulk whatever. Any questions on this before we move on to secondary active transport? Any questions?
Hello, Prof.
Yes, sir.
I've got a question.
Yes, sir.
Does bulk transport require energy?
Does it require energy? You saw that there was ATP when it is coming for exocytosis. You saw ATP is required. When the vesicle comes in and it is carrying a combination of proteins, just before it fuses with the plasma membrane, where I showed you it does the kiss and run, it requires ATP at that point. Only in exocytosis, but not in endocytosis. Are we clear?
Yes, you are clear.
Okay. Anybody else? Anybody else? Okay, hopefully we've refreshed ourselves on the last couple of lectures, which have probably been a total nightmare. So let's go back to the transport of small molecules. I told you that we were going to go back to secondary active transport, and now we're going to go into that in quite a bit of detail. In some cases, I'm going to introduce some issues which we'll be looking at in more details later on. But in the meantime, I'll try and mix it up as well as I can. And this is based on where I saw your cousins in previous years having issues in terms of understanding. So the internal composition of the cell is maintained because the plasma membrane is selectively permeable to small molecules. Okay? Most biological molecules we saw are unable to diffuse the phospholipid bilayer, so the plasma membrane will form a barrier, and this is the barrier that blocks free exchange of molecules between the cytoplasm and the external environment of the cell. So specific transport proteins, which we met before, carrier proteins and channel proteins, will then mediate the selective passage of small molecules across the membrane, and in doing so, they therefore allow the cell to control the composition of the cytoplasm inside. So I'll give you an example. This is the lumen. This is, for example, in your gut. So your food is coming in. Here's your glucose and broken down sugars, carbohydrates, which will come in. And remember, we said glucose does not cross the plasma membrane just like that. right? So you have the molecules, the carrier proteins, the transporters sitting on the plasma membrane. And then you also have the ions. So when they tell you to eat a balanced diet, don't joke. You know, the sodiums, the potassium, all the minerals in your diet are very important. So glucose will be moved in from the lumen into the cell. This is the apical side. Those of you who did cell biology with me will recall that these are called tight junctions, and these tight junctions are the ones that control seepage of materials in between cells. This is the interstitial fluid between two different cells. So you don't want these materials seeping in between two cells. If you give me 1 second, I'll grab a So I beg your pardon. So the glucose or materials, large molecules or ions can come in through the apical side, and within the cell, they also are controlled in how they move from inside the cell to outside the cell. You know that lining your gut, you have that layer of epithelium, then you have longitudinal muscle, then you have circular muscle, and inner space in that is blood vessels, right? So when you eat your lunch or breakfast or tea or whatever, the molecules which have been broken down by whatever you did in physiology come in, and they are controlled in terms of how they end up in the blood. They don't just, by osmosis just go in. So you have potassium ions which go through the potassium channels, remember those? Or you have the potassium going in against the concentration gradient, which requires ATP, because this is unfavorable. You don't get from low to high. And when that happens, you need ATP. And then you also have sodium. So this is the sodium potassium pump, or the sodium potassium pump, which then will cause these sodium ions to go all the way to the blood circulation. Okay? Now we are beginning to combine these things. So secondary active transport is a form of active transport which crosses biological membranes. A transporter or protein is the one that is responsible for coupling the movement of an ion, and this is typically either sodium or potassium, down its electrochemical gradient to the uphill movement of another molecule ion against concentration or electrochemical gradient, which we talked about in the last lecture. So energy, which is stored in the electrochemical gradient of an ion, is used to drive the transport of another solute against its concentration gradient or electrochemical gradient, which I just showed you in the previous slide. The ion, which is moving down, its electrochemical gradient is referred to as the driving ion, because it is movement of this ion that will drive the uphill movement of another ion or molecule, which is the driven molecule. So in here, potassium is being driven uphill, and sodium also being driven downhill because of the higher concentration in the interstitial fluid. So what are these driving forces which act on molecules? Molecules will move from an area of high energy to low energy. And there are three forces that cause molecules to move in this direction: chemical, electrical, or electro and chemical. So a combination of the two. This force is directly proportional to the concentration gradient. The greater the gradient, the greater the force. If there are more than one kind of molecules across a cell membrane, then each of these molecules has its own concentration gradient. So sodium to sodium, potassium to potassium, calcium to calcium, chloride to chloride. So you don't mix and match. So it has its own concentration gradient or its own chemical driving force because they have different charges. So you have chloride which are negatively charged, you have sodium which is positively charged. So what is this chemical driving force? In here you have a high concentration as depicted by the many red dots, and the chemical driving force is outside. You can also have a chemical driving force from the extracellular side, where the red dots are more on the extracellular side and they're less on the intracellular, and the chemical driving force can be in this direction. So the chemical driving force can either be in the extracellular direction or intracellular direction, depending on the concentration of that particular ion, wherever it is. So when a molecule exists in different concentration on either side of the membrane, a concentration gradient is said to exist across the membrane. Oh, I forgot to mention. So this here, these two lines here depict the lipid bilayer or the plasma membrane. So when we refer to membrane, from now on, we are talking about the lipid bilayer or the plasma membrane. So this concentration gradient is said to exist across the membrane because of the differences in concentration either intracellularly or extracellularly. Delta C, I don't know what happened here. It's not a box C, but it's delta C, symbolizes this difference in concentrations. So molecules will move because of their net thermal motion from an area of higher concentration to an area of lower concentration. This is nothing new to you because I think you did it in high school in osmosis. So the net force which drives molecules down concentration gradient is called the chemical driving force. Molecules move from a region of high concentration spontaneously. In other words, this happens because it results in release of energy. And you know that it moves spontaneously so that it can create equilibrium. So it will obviously move from high to low to create that equilibrium. Molecules in this situation are described as going down their concentration gradient. So it's moving from high to low, and therefore the molecule is moving down its own concentration gradient. In contrast, with molecules which go from low concentration to high concentration require energy, like ATP, right? Molecules are then described as going up their concentration gradient. Are we clear on that point? Because we'll be talking about down gradients, up gradients, and all whatnot. Would you like a minute to copy this? Yes, no. Type yes or no. I can give you a minute to copy this because you really need it for the rest of the course. Otieno wants to copy it. Okay, Laura.
Let's give us 1 minute.
potential is measured in millivolts. So basically, I know I saw some of my colleagues in the UK trying to measure the charges between here. So they would take a pasture pipette and put it on a Bunsen burner and make an electrode, which is put it on a Bunsen burner and stretch it so it's a tip. So they put it on one side and then take another one and put it on another side and then connect those to a voltimeter and actually add chemicals or add drugs to look at what happens to the plasma membrane potential when they added different solutions, either extracellularly or intracellularly. Some really cool, fancy projects. So the size of the membrane potential is measured in millivolts, and that, of course, is 1,000th of a volt. I mean, it's really little, and it's proportional to the number of opposite charges separated by the membrane. So by convention, the sign of membrane potential refers to the net charge of ions inside the cell compared to outside the cell. In the resting cell, that is if the cell is not doing anything, there are more anions within the cell inside the intracellular cytoplasm compared to outside the cell. In simple English, it's more negative inside than it is outside. Hence, the potential is negative. For most cells, the membrane potential is around negative 70 millivolts. So this is on average, and this is for most cells. We are going to be talking about nerve cells in detail, and we'll be talking about the resting potential, minus 70 millivolts. And God help you if you ask me, why is the resting potential minus 70 millivolts? Those of you who've been through this class before, here is where you used to get stuck. Hence why I've given this class this slide. So I will give you time to copy this one. If you have any questions, please type in, and I don't want to lose anybody as we go along. It's much better to understand it at this stage than to get lost. Any questions? While people are copying. I know some of you take screenshots. If you share those screenshots, be careful of copyrights. Wangeshi, are you there? Many of you are on mobile. Is Wangeshi there? Yes. Wendy? Do you understand? Wendy Guantai. He logged in and went for a walk. Wendy Guantai. Absent. Oh, she's there. Somebody called her.
Hello, Prof.
Yes, sir.
I've got a question.
Go ahead.
You said that the movement of the molecules from high concentration to low concentrations leads to there is release of energy. So I was asking myself, where does that energy comes from?
The movement of the ions.
Okay, thank you.
You're welcome. Anybody else? Okay, so the chemical driving forces in scenario in living cells. So here again is the plasma membrane, and here is the electrical driving force. You can see the positive ion trying to go in, or the opposite negative ion electrical driving force going out. For a negative ion to go out of the membrane when it is surrounded by positive charge will be a Herculean task, because these instead attract rather than repel. So for this to pass through would be tough. So this is called the chemical driving force. And this is what happens in living cells. Positive charges going intracellularly past the negative charges on the membrane side of the intracellular space, and vice versa. From the negative charges going out past positive charges on the membrane on the outside. So we talked about the combination, the electrical and the chemical combined, called the electrochemical driving force. And this, of course, as the name suggests, is the total forces acting upon ions across the membrane, is a combination of both chemical and electrical forces, and is referred to as the electrochemical driving force. The net direction of this force is equal to the sum of both forces. So it's equal to the sum of electrical and also the chemical. Just add them up. If the chemical and electrical driving forces are in opposite direction, the point at which each equals the other is called the equilibrium potential. The equilibrium potential of an ion will depend upon its concentration gradient and, of course, its valence, which is the charge. The equilibrium potential can be determined using what we have seen before, the Nernst equation. That is the concentration outside and the concentration inside. Remember, we used that Nernst equation for that. So I'll let you copy this also if you don't have any questions. 2 minutes. David Kabuyu Mwangi, are you present?
Hello.
Do you understand this, Mr. Mwangi? Mr. Mwangi, is that clear?
Yes,
Okay. Daniel, you might be sharing your secrets with the whole class. Can you mute your mic, please?
Hello, bro. Hello.
Hello?
Yes, I have a question.
Yes, go ahead.
There is a time I asked you a question about the TCL that's a component in the bank. You told me when we reach the secondary driving forces, the secondary membrane, you shall tell us the answer for the question.
What was the question?
So what is the
Repeat again.
The question was how the TCL, that's the marijuana content, how it moves to the brain, and how it makes someone to be high.
We are going to get that to that. Remember, when you look at your course outline, you have serotonins, catecholamines, and all those things. Hello? Hello?
Yes.
So we are going to talk about how that happens when we reach serotonins and catecholamines. However, you so far we've talked about how big molecules move in the cell, right? Glucose. I showed you glucose comes in and goes and enters the blood, right?
Yes.
And once it enters the blood, it travels to the brain, right?
Yes.
And basically, I think you ingest marijuana. It's not marijuana. Marijuana. The J is silent. You ingest marijuana through your face, either. Is it smoked or chewed?
Through the lungs. We smoke.
You smoke it.
Through the lungs.
Okay, through the lungs. So you know about the interaction of the alveoli with the capillaries, right?
Yes.
And the capillaries take up everything, including air from the alveoli.
Yeah.
Those molecules, the catecholamines and those nasty molecules which are in your marijuana, are then taken up into the alveoli through secondary active transport, just like glucose, because they are large molecules. They don't diffuse.
Yes.
Into your blood circulation.
Yes.
And then from your lungs, where does the blood go? To the heart.
To the heart.
And then from the heart, it is pumped where?
Brain.
And then in the brain, it crosses again by active transport into the brain cells.
Yeah.
And then inside the brain cells, it makes you a zombie.
Okay, thank you.
How it makes you a zombie? Again, in the brain cells, which are also epithelial cells, it passes through the apical side because it has to pass the way it is supposed to the blood vessels. I will then tell you now exactly what those chemical compositions do inside the brain. Okay?
Yes, okay, thank you.
Okay, remember that. Oh my God. Are you one of those people who visit the guys who sell one bunch of sukuma the whole day behind Tuk? Or you know those guys? Daniel.
I just know them. I know them. They're just my friends.
They're your friends?
Yes, they're just my friends, but I don't taste.
You don't taste?
Yes.
But you stand around them.
Yes, I'm always eager to know what makes them to be high. That's my question.
Hopefully you don't stand around them because you can't block it.
Yes. Yeah, I know I can be a passive smoker, so I avoid the distance. I do social distance.
There's no social distancing with this thing. Is it 1 meter, 2 meter, 10 meters?
Just 10 meters away from them, completely away from them.
All right, so here's an example. Potassium, chemical driving force going out. You know, potassium ions are positively charged in the membrane here. You have negative charges opposed to the plasma membrane, and then outside the plasma membrane, you have positive charge. So electrical driving force, because of the charge, they're driven in, and the chemical driving force, because of the charge on the potassium, they're driven out. So both can actually happen, and that is when you get electromotive or electrochemical forces combined. So secondary active transport can also involve coupling of ions and also pumps, which actually move ions from one place to the other. We did a little bit of this. Pumps, we talked about the integral membrane proteins that act as pumps. So we're going to look a little bit into more detail and distinguish between the coupled transport and, of course, the role of pumps in moving molecules. So in ion coupled transport, Coupling between the driving and the driven species is obligatory. Both the driving and the driven species must be bound to the transporter for translocation across the membrane to occur. And remember, this transporter was a channel protein. Remember the transportation? And unlike primary active transport, in which ATP is hydrolyzed. This provides free energy which is needed to move solids against a concentration gradient. In secondary active transport, the free energy needed to perform active transport is provided by the concentration gradient. I think somebody asked me where the free energy is from, and the concentration gradient of the driving ion. So I said the ions itself. So this is what provides the energy for movement. Ion pumps, on the other hand, is a whole different kettle of wax. The existence and maintenance of the concentration gradient of driving ions is accomplished by primary active transporters. This is just a fancy name for pumps. Sodium serves as the driving ion in many, but not all, secondary active transporters which are located in the plasma membrane of various cells. There is a steep sodium concentration gradient across the plasma membrane that is maintained by the sodium, potassium, ATPase. So this is a combination. It's a protein that binds sodium, potassium, and the enzyme that is used to break down ATP. And this is called the sodium potassium ATPase pump. Okay? I think you need to take this because we'll be talking about the sodium potassium ATPase pump for quite a bit later on. Let's lay the groundwork. Away from it. The extracellular sodium concentration, which is the concentration of sodium outside. When you see this in future, this means the concentration of whatever ion outside is around 145 millimolars, and the intracellular sodium concentration inside is around 15 millimolar. The free energy stored in this gradient is used by sodium coupled transporters to drive the uphill movement of substrates. And why do we say substrates? Because remember, there's ATPase which is involved also. So the ATPases also act on the substrate. So the free energy, the difference in 145 and 15 is what creates the energy gradient between the extracellular side and the intracellular side of mammalian cells. And that's why sometimes when somebody is unwell, one of the first things you see in hospital is that they quickly insert an IV with saline. And that saline contains sodium along with other ions. It is really important to have this balance of extracellular and intracellular sodium concentration. So that just this slide in itself tells you why people get IVs, so that you can balance this concentration in that patient before you start doing other things. Obviously, you have heard the layman's term, ameenda hospitali amewekwa maji. Nope, it's not maji. It is these ions, and these ions are the ion coupled transport in the secondary active transport. So, there are two types of secondary active transport. One is co-transport, where molecules are moved along together. Remember antiport, symport, and uniport, and exchange. There are also two variations of secondary active transport. Co-transport, which we now know as symport, and exchange, which we also know as antiport. And these terms are used interchangeably sometimes. That's why I needed you to learn how to define these. And by now you should know what antiport is. The picture should come in your brain, and symport should come in your brain. That's why I started by that. The transport proteins responsible for secondary active transport are called as secondary active transporters and are specifically referred to as the co-transporters or the exchanges. Sodium-coupled transporters and proton-coupled transporters and exchanges. Sodium is the driving ion for many co-transporters and exchanges. Therefore, these transport proteins may also be referred to as sodium-coupled transporters. Similarly, there are many examples of proton-coupled co-transporters and exchanges. So the key driving ion is sodium. When one ion is moved, chances are sodium is the driving ion. In other situations, you'll find that it is potassium ions. And that's why I showed you that as the very first example of transportation across the plasma membrane. So, we're not going to look at everything here, but I just wanted to show you some examples. That's sodium, and that's sodium. This is co-transport, X, and exchange, Y. The exchange is antiport. The co-transport is symport. In co-transport, sodium moves in and molecule X is also moved in. In antiport or exchange, molecule Y moves out and sodium moves in. Okay? Remember, in primary active transport, remember we did this? Sodium moves out, requires ATP, and this ATP broken down to ADP will cause potassium to be moved in. So this is the distinguishing feature between co-transport, symport, and exchange, antiport, and primary active transport. X and Y molecules are being moved by secondary active transport. So I'll let you copy this because it's also a baseline. You can also find quite a lot of resource here. You can copy this, www.physiologyweb.com. You'll find a lot more of these, but I've just used one to explain. All right? So copy this if you wish. Are driving forces applicable in all cells regardless of their age? Yes, as long as a cell is alive. Valerie, that's a good question. If a cell is dead, driving forces are not there, and at that time, there's a process which starts getting into play. Do you know what that force is called? That action. Once cells are no longer viable. Valerie? Anybody? Can anybody contribute? Have you heard of apoptosis? Or Have you heard of that? Apoptosis. Anybody? So obviously, driving forces are not as efficient as you get older. That's why you find in older people's skin, they don't look very, very good. Yeah, so obviously, Diana, a lot of these things, what's the first thing that happens during apoptosis? That happens. Okay, there are several things that happen during apoptosis, but the last one is fragmentation of the plasma membrane, right? So when the plasma membrane is fragmented, all these things that happen on the plasma membrane are not going to happen. Okay? Valerie, are you satisfied? Okay. So secondary active transporters may also be localized to the membrane of internal organelle. Robert Muka Amatete, can you lower your hands, please? They're also localized to the membrane of internal organelles. Remember, we said that internal organelles such as mitochondria, the Golgi, also are enclosed in a lipid bilayer of a membrane. So if they have a lipid bilayer of a membrane, then you will have secondary active transporters. And these are very high in mitochondria, for example, because you know mitochondria is very, very highly involved in metabolism. So you'll have a lot of shuttling of ions and molecules across the membrane of mitochondria. So in vesicular membranes, these are membranes which are inside the cell and not on the plasma membrane of the cell. So secondary active transporters may also be localized in the membrane of internal organelles. An example is protons and neurotransmitter exchangers use their proton electrochemical gradient or proton electromotive force, also known as EMF, which is established by the voltage type proton ATPase to drive neurotransmitter molecules against their concentration gradient into the lumen of synaptic vesicles. Neurotransmitters, protons, ATP, AGP, and pyrophosphate all are involved in transport coupling stoichiometry that determines the effectiveness of substrate transport against a concentration gradient. So where you find a lot of active movement, ATP, protons, and ATP, chances are you'll need a coupling stoichiometry, that is how many molecules will be needed. So you can have two sodiums moved against one iodide, and we're going to talk in detail about some of those scenarios coming up pretty shortly here. So an example is the neuromuscular junction. This is a nerve axis. For any given transporter, the coupling stoichiometry is usually a fixed ratio. So in this case, it is 2:1. A higher ion substrate coupling ratio, for example 3:1 versus 2:1, leads to a significantly higher concentrative capacity of transport. So if you had 3:1, you'd get a higher concentrative capacity of transport. A consequence of the ion substrate coupling ratio is that activity of some co-transporters and exchanges leads to translocation of net charge across the membrane. A transporter that leads to the net translocation of charge across the membrane is called electrogenic, because basically it's moving more sodium from one compartment to the other than was actually there initially, concentrating it inside. So co-transporters can also be electroneutral in addition to being electrogenic. For example, when you have a coupling stoichiometry of two ions, sodium ions, to one glucose or galactose molecule, the sodium glucose co-transport by sodium glucose transporter 1, this is SGLT1, which I know many of you have measured in labs, represents an electrogenic process because you have a net translocation of charge across the membrane. Such charge movements across the membrane lead to small electrical currents, and therefore you have electrophysiological methods which can be used to measure the activity of electrogenic transporters. When no net charge is transported per membrane cycle, the process is said to be electroneutral, and I'll show you some examples of that in a moment. First, co-transport proteins are found in many different cells and tissues, and they perform quite a number of
Unajua unaweza type na hii tena. Unaweza type na mouse.
Maconnect na ya OTG.
Na nimeconnect na OTG na inafanya kama mouse.
Infection zako. Na infection ni.
Akili muhimu.
I got chucked. I seem to have managed to come back with minutes to spare. We'll take every minute we can of this lecture. Maida, mwalimu yuko. The session is not over. I got chucked out. So let's look at the sodium glucose transporter, SGLT1. And the sodium glucose transporter is found in the apical membrane of epithelial cells of small intestines and the renal proximal tubules. Remember, I gave you an assignment which is due in a week. So, but that was about something else. So the sodium glucose transporter SGLT1 is found in apical membrane of epithelial cells of the small intestines and the renal proximal tubules. It utilizes sodium electrochemical gradient to drive the uphill transport of glucose into the cell. Uphill meaning from low concentration to high concentration. So this is what the SGLT transporter is there, sitting on the plasma, on the lipid bilayer of the cells, of the epithelial cells. And 2 sodium ions moved in to the cytoplasmic space along with one glucose molecules, hence the name co-transporter. If they were in opposite direction, it would be called an exchanger. Okay, so that is one example of what we've been talking about in terms of co-transporters. The coupling stoichiometry of SGLT, the iron substrate coupling stoichiometry of SGLT is 2 sodium ions to every glucose or galactose molecule. Therefore, SGLT1 is an electrogenic transporter. In addition, SGLT1 also recognizes galactose, as a substrate. And SGLT is really important because its function is crucial to dietary glucose and galactose absorption. Oh, come on. Really? Really? It's really important for galactose absorption in the small intestine and reabsorption of filtered glucose and galactose in the proximal tubule of kidney nephrons. So this is a live example of how transporters, sodium iron substrate coupling is really important for the physiology of glucose galactose absorption and also filtration in kidney nephrons. So this is a good example. Another example is called the sodium. I'll let you copy this one. You can copy this one first. And then I'll let you copy the next slide because this is a really important foundational molecule for you to be able to understand the others. We have about 15 minutes, ladies and gentlemen, so let's take everything we can since we've had a peaceful ride so far. If anyone has questions while others are copying, you can go ahead and ask. Let's deal with those as we go along. Mike, we are missing some students. I only have 31 on record, so tell them I'm actually downloading the user attendance from a screenshot, so I still have my attendance sheet. So please let those who know who didn't attend that they're going to have a rough ride if they've missed this basic foundational thing. Thank you. Thank you. Phosphate transporter is found in the apical membrane of epithelial cells of the small intestine and the renal proximal tubules, and it also utilizes the sodium electrochemical gradient to drive uphill transport of inorganic phosphate into the cell. So for every transport cycle, sodium inorganic phosphate, which is called 2N2B, but this is not important. This is what is important. Couples inward translocation of 3 sodium ions and 1 divalent inorganic phosphate. Okay? So the sodium inorganic phosphate types 2A and 2B are electrogenic transporters. There's a whole bunch of others, but I'm just generalizing for this one. A third type, sodium inorganic phosphate 2C, is electroneutral, which couples 2 sodium ions to a divalent inorganic phosphate. So that is 2 to 2, therefore you get electroneutral. So you can copy this slide and we'll be done after I show you the image in the next slide. Ameline, do you have a question? I see a raised hand. Ameline, do you have a question? I see a raised hand. Ameline Emali. Okay. So there's a whole series of them. Maybe I'll print this slide and maybe give it to Michael. You can pick it up from my office. Mike, you can pick it up from my office.
Yes. When should I pick it?
Well,
Tomorrow.
no, I can't do that tomorrow. You know the network in my office is rubbish.
Okay, so I can pick it on Friday.
Yeah. Am I going to be able to be able? No, let's do it Monday. Okay. Because I don't know if I'll Monday on Friday. So sodium glucose water, sodium and 1 glucose, coupling stoichiometry 2:1, sodium phosphate co-transporter 2A and 2B, 3 sodiums to 1 inorganic phosphate, also moving it inside, sodium iodide, symporter, 2 sodiums to 1 iodide, and they're moving them in the similar direction, and it's abbreviated NIS. Sodium, potassium, chloride, co-transporter, and this is abbreviated NKCC. So 1 sodium, 2 chloride, and 1 potassium. So you can see it's 1, 2, 1, coupling stoichiometry. Sodium, chloride, co-transporter, referred to as NCC, 1 sodium to 1 chloride, coupling stoichiometry of 1:1 ratio, and moving the molecules in, so it's a co-transport, and then sodium chloride co-transporter moving from inside, potassium chloride, moving potassium out and 1 chloride out, coupling stoichiometry of 1:1. Again, I said this is not in the books that I have referenced for you, but I thought these were really cool images from physiologyweb.com, which also has some very interesting resources. If you want, you can go and learn a lot of these things over there and kind of just understand it better. So if you have any questions, let me know. We have a minute. We have a minute for any questions. Minute. Do you understand? Are you understanding, or have I lost anybody? No? Okay. I think we are good for today. I think we made some good progress. We are going to pretty much be on course after tomorrow's lecture, which I have to make this afternoon. Good heavens. Any questions? Okay. Bibi, you're speaking for others. Do they understand? Okay, yes, It's not it's I am. Vivian is typing. Anybody else? Anybody with a burning question? We're going to be chucked just now. Okay, Vivian, good. Thank you for the feedback. So, Odhiambo, you've been AWOL. Good to see you. Daniel, bye. All right, tomorrow. I'll send Mike a link, same system. I'll try and set it up just now.